on our work and our faith in our profession, but particularly now I'll be talking about genetics. I'm quite nervous because I'm talking. I don't know what level I will talk, but I want to go to the basic as much as possible. So I'll be talking more on about my genetics and my faith. I want to share my video. I hope I'll be doing it good this time. Okay. I think I'm okay. So, but it's not on the right page. Sorry. At least you know already what I'll be talking about. I titled it My Genetics and My Faith, and I'll be talking more on how I appreciate my field of expertise in the science of genetics, particularly crop genetics or crop breeding, which I have been doing it for the last more than 34 years. I just want to show you that I have been, after my graduation, I joined the International Rice Research Institute as a researcher, and I climbed up from the ranks to be a senior scientist. And in one point of time, I was based in Africa to work as a rice breeder for Africa, and I developed several varieties. I trained several young scientists, and I hope I have done good enough in my contribution for the science of genetics for rice. After 29 years, I went to the industry. I joined a private company, and I work on corn, sweet corn and waxy corn. If I describe what I'm doing there, I I I play with colors and sweetness on corn. So that's why I have different colors and I have different sweetness of corn. I work on waxy corn and I work on sweet corn. So in the field of genetics, I just want to emphasize that genetics is actually is the science of variation and heritability. So most of the things around us is genetics. You could see in your left side, you could see your neighbor, you could see your family, you could see plants, you could see animals, pharmaceuticals, even the pandemic that we have now, you could see all in genetics. So I will try to explain it in a layman's term, but I won't guarantee that I will explain it enough because it took me how many years? 4 plus 5, maybe almost 19 years of schooling to study the field of genetics, but I'm still learning up to now. So with this one, it's, as I told you, genetics is study of variability, why things are changing. At the same time, why it is inherited from one generation to the next. So this is genetics. So you could see here an example of my, I think they are my kids few years back or more than few years back. So you could see the variations there, but they are genetically, they are very similar, but you could see still the variation. If you will not go into the details, you will see they are coming from the different families, but these are just example of, you can see variation, and we will study how it is changing, why they are different, but and how have been, what are the traits that they inherit together. So that's genetics. If we go to the definition of genetics, let's go to the definition, because before we say, as Aristotle always mentioned, before we study anything, we have to define our terms. We have to define everything. So you'll start with chromosome. Part of the cell is the chromosome, which is the very important part of genetics. So chromosome, if I describe it, by the way, in Tagalog, they call it kulay lawas, because chromosome, it means color, a color of a body, because that's the part of the cell. When you dye it, it will produce colors. That's why it could be seen in a microscope. So chromosome is like a book. If I describe a chromosome, it's like a book. And each individual chromosome being a chapter. So if the whole genome is a book, then you have the chromosomes are the chapters. And if you say genes, what are genes? Genes are, these are individual instructions for making proteins. These are like, genes are like, it's like piano keys. You can create chords, melodies, or infinite varieties of combinations. But one gene is just nothing. It will just do, 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 do. But if you combine several genes, it could create melody. It could create something. That's why human being, animals, plants, individual species, and individual bacteria are functioning together to make them work. They have a group of genes that work together in harmony to create life. Okay, so genes are very important. This is the thing that we are studying. If you say a human being, before they say the human being has 100,000 different kinds of genes. But after careful study, they now say it's about 35,000 to 40,000 genes. But individual genes will not work alone. It is a combination of several genes and they have to work in harmony. So it's a very complicated thing. You could see just looking at the genes, seeing at the genes, you will see life, you will see how it is being created, how it's being replicated, and how it is passed from generation to generation. So it's so completed, it's so beautiful. You will just be amazed how God made this one. And for centuries, we are studying genes, and up to now, we still don't understand all of it, which I'll be going to the other things, other explanation later. Codons. These are the genetic codons. These are the codes. These are the words by which instructions are written. So it's the code. It's the genetic code, which I'll be talking later. AUG, the three codes, the triple code that codes a single amino acid. And this amino acid will combine to each other to produce a protein. And this protein will interact to produce something functional. Okay? And bases, these are individual letters. There are 4 bases. You said A, T, G, C. Adenine, thymine, guanine, and cytosine. So these are 4 nitrogen bases. Even by defining all these 4, I need one day to explain this. But just bear with me. Just ask me specific questions if you want more elaboration this one. Now, if you say genome, you have now genes, you have codons, you have genome. Genome are instruction manual. This is how to assemble the body. These are the different genes, group of genes of the body. It's an inventory of parts of a living body. It tells us what we are made of, but does not say how it works. So parang kuan lang siya, instruction manual. Ito ang spark plug, ito ang different parts ng car, but it will not say how it functions. So it's just an inventory. And now you go, what we need is the operating manual, how it works. That is where genome, proteome come in. I'll not be going to the details of that. Proteome is the library information that create proteins, much more complicated than genome. So that's how the definition is. If you go to the next slide, this is just a caricature to make it more friendly to look like, but it's so complicated. You could see there the cell, and the middle of the cell you have the nucleus, and in the nucleus at the middle of the nucleus is a nucleolus. That's where the chromosomes are coming in. Okay, that's why it's the most darkest part of that's where the chromosome is. Okay, if you go to the details of that, you could see the chromosome. And the chromosome is intertwined together to make all this DNA, deoxyribose nucleic acid. And you will see in one cell alone, if you will stretch the chromosome, if you will stretch this chromosome, it is actually 2 meters. It's 2 meters. So in your body, you have billions, trillions of cells. Actually, if you connect them together, it will travel, it's more than 3 billion miles. So you could really, it will connect from Earth to the moon and back to the moon and back again, maybe 20 times. So that's how amazing our DNA is. Okay, so I will not overwhelm you, but that's how amazing and these are functioning in a harmonious manner. So it's really beautiful. You will be just amazed of nature. We are just talking about the cell and its function. This is just the length. So you could see who is our engineer in our life. If you go to the next slide, this is the central dogma. This is where we study everything. You have the deoxyribose nucleic acid. This is the DNA, and it could do replication. It could replicate itself in split seconds. You could produce a replication of your DNA. And if you go further, it will produce an RNA. This is the ribonucleic acid. And this is the one that will change. This will translate into an amino acid or becomes a protein. Because the DNA could not make into an amino acid right away, you need an interpreter. It's like the DNA could not produce an amino acid or a protein. A protein is a group of amino acids. So it's like DNA could not communicate to protein, so you need an RNA. So the RNA is the interpreter. It's like maybe this is a Spanish speaking RNA and the other one is a Tagalog speaking amino acid. So you could not connect. So you need an RNA, which may be an English language to translate the Tagalog or the Spanish to Tagalog. So this is how it works to make a functional enzyme or a protein from amino acid, from DNA to RNA to amino acid to protein, until the protein will combine together and produce a functional organ. So it's so beautiful and it's so complicated that once you elucidate how these things work, you will just amazed how these things happen. And what happened now is many scientists started to discover it. And once they discover it, they feel they are God. We are just discovering a small portion of God's creation. Now, if you see, this is another complicated thing. This is the genetic code. This is from RNA. It will translate into an amino acid. So every codes are there. As I mentioned, there are 4 codes. In computer, you have 010101 to create a program. It's a combination of 010101 to create something. But for the genes, for life, for the DNA, you need 4 bases and 4 letters, and 3 tricodons will produce an amino acid to produce a protein. Okay? I will leave it that way. That's the complication of this. So it's a very specific activity of the cell to create a new protein that is very specific to assemble together to create life. Okay, so this is how the things are happening. Okay, so if you have those things, you will create a lot of beautiful things out of those genetic events happening in our life. You could see an eggplant, you could see a flower, you could see a human being, you could get even a black tomato, all types of tomato, even in rice. Do you believe that we have more than 130,000 different kinds of rice? That's only the one cultivated, or that is one collected. it must go up to 200,000 if you collect all different kinds of rice. That's rice alone. How about tomato? How about the flowers? How about the bacteria? How about all those stuff? So you can see now how beautiful is our creation. Up to you could see all these beautiful things created by developing, by harnessing the different variation and select the best uh, varieties or species for human needs. That's where plant breeding will come in. That's where biotechnology will come in. Okay, so this is how it goes. So you have a DNA, and in a DNA has the genetic code of life. And it is very precise that the different combination will create human being. A different combination, but still A, T, C, G. Only 4. It's a combination of the different combinations, and it will create a human being. It will create a tomato. It will create a bacteria. It will create a fish by only these 4 letters. So what does it mean? If you have 4 letters, from bacteria or living organism, from bacteria to plants to animals to human beings, only 4. So what does it mean? For me, it means there's only one creator. There's only one engineer. There's only one geneticist who made this one, because the template is A, T, C, G. It's like the computer, 0101. There's the first one who discovered it, and it branched out to make it different programs. So for this one, there's someone up there who invented this one, who have this one, who created this one, and made everything like what we have now. If you see now how I'm now going to the field of plant breeding, which is the science of applying genetic principles to improve plants for human use. Just to give you an example, before corn, because of the improvement of corn from teosinte, it becomes a corn. So that's how plants have changed. But these are very related crops. You could see here how hybridization by human intervention, we were able to create new variations by crossing them together and creating new combination to create new varieties. So that's where my field of expertise come in, which I have been working with the International Rice Research Institute for 29 years. And the team were able to develop some crops that could, by selection, by genetics, by studying molecular genetics, by applying it, and the team were able to create a rice that could tolerate under flooding condition. Just like this example, you see they are planting the same crop, but the other crop on the left side was as submergence tolerance after flooding. The rest of the neighboring crops died, the crops, but the other crop, which is a product of plant breeding, survived because it has the genes for survival under drought condition. And this is my work, which I'm proud of, where we develop salt-tolerant varieties. This is a picture of, on the right side of the farmer in Bangladesh. He planted a variety which is salt-tolerant. Under salt condition, you can see his variety is already dying, but the salt-tolerant variety on the right side is still surviving. Same is true with this one, the salt-tolerant variety we developed at the left side. At the right side, this is a product of genetics, of plant breeding. And the other side is a salt-affected area. This is, they are producing salt here on the right, on the left side. On the right side, which is also salty, but we were able to plant rice there because it has the gene for salt tolerance. So this is through genetic manipulation, we were able to develop a better crop. So these are the different combination of genes. They are very related, but they could combine differently and produce different kinds of grains, of different kinds of crops. Like this one, these are very related. The wild mustard plant, this is the brassicas. And if you have different combination, you could create the the kohlrabi, kohlrabi, you could create a kale, you could create a broccoli, Brussels, cabbage, and cauliflower. They are actually, they could cross together, interbreed together. So you could just appreciate the variations. And if you want, you could even have a banana with many, many, many fruits. And as I told you, I'll be talking about the biotechnology, which is also a product of genetics, and have the grafting, tissue culture. These are all biotechnology. But now we are doing about crop modification, which is very popular now and also very controversial. So we do a lot of crossbreeding, mutation, increasing the ploidy level to have a bigger fruits, and without seeds. You could use a protoplast fusion. You could fuse the different protoplasts to develop, to create a new product. We have transgenic, which is the GMO. This is adding a gene, additional gene from other species to another species through gene splicing. And latest now, which is technically not a GMO, but we use gene editing, directly editing the, as I mentioned, these 4 letters, ATCG. By manipulating the combination, we could create, by editing it, we could create new products. So genetic engineering means to a specific, refers to a specific gene transfer from the same or related or unrelated species, resulting in a gene fragment modification and combination. So you could see here that the genetic engineering actually is to produce a new trait in an organism by combining unrelated traits. Okay, getting the gene from daffodil, putting it to rice to produce a golden rice. And they got a gene from corn and put it in rice. You want to get a gene from maybe a fish and put it in a plant. That's possible now. You are just putting a very specific gene. Okay, so let's go to the hot topic, which is the GMO. So what are GMO? Genetically modified organisms are plants or animals where DNA has been altered. There should be altered through insertion or deletion or rearrangement that does not occur naturally by mating or by natural recombination. So that's why it's GMO. It's unnatural. So it's a way of inserting genes from unrelated things to create something new. Okay. So why GMO was produced? So these are the perceived advantages to producers and consumers. Because of GMO, the food prices have been lowered because you could produce a lot with a higher yield, better quality, and less of the pesticides and other inputs. It has insect resistance, virus resistance, herbicides tolerance, nutritional value, and it produces higher yields. So those are just examples of the advantage. And up to now, just to give you an information, we have now more than 18 million farmers are using GMO crops from 28 countries, and about almost 200 million hectares now are using GMO. And it has been increasing all the time. So all the clothes, most of the clothes they're using are from cotton, GMO cotton. The milk, soybeans are all 90% or 80% are GMO. The oil, canola oil that we are taking, even the corn, it's all GMO. Even in the Philippines, we're using GMO corn. And just to give you an idea what we have in the Philippines, this is the biotech crops in the pipelines in the Philippines. These are GMO. We have the BT eggplant. This is an eggplant. This is resistant to the insect. that makes the eggplant hard, and you could see a lot of worms inside. BT cotton, and also the papaya that will delay ripening so that you could transport it. You don't need to eat it it will ripen a longer time for transportation, so that is good. And the latest is the golden rice, which high in vitamin A, provitamin A for human health. We have another work that Bill Gates was funding it for several years now, more than almost 20 years. This is changing the lifestyle of rice to behave like corn. So this is the C4 rice. I'll just give you an idea that they want rice to grow without water or less water, not standing water. So we could lessen the production of rice with less water. At the same time, it has higher yield and efficient in the utilization of nitrogen. This one, I'll just show you this one. This is not a product of genetics. You could see a square watermelon, or you have a heart-shaped watermelon, but these are just a manipulation of the container. So once the fruits are getting bigger, still small, you put it in a round or a box type container, and it will form itself in that container. Then after that, you open it up and it will look like this. The other one is a heart shape. So you could see some manipulation, but not genetic, but you could create something like this. Sometimes these are not GMO. Okay, so if you want, you could create something like this, but this is just an example of a fruit where you have everything. You have already halo-halo. You need only ice and even the milk, coconut milk is already there. So you have already halo-halo. You need only ice and sugar. And for animals, we have genetically engineered animals. The first one was Dolly. This is a sheep, first genetically engineered animal. We have a lamb. They call it Polly, that produce a factor 9 protein in milk. We have Diane and Peter. We have Diane and Peter. These are sheep. And we have Webster and Peter. And in the next slide, you have genetically engineered primates cloned from embryonic cells, and Niti and Dito. We have Andy, we have Senna, we have cloned pig that has genome of black skin pig. So these are just example of manipulated genetic manipulation to produce a new creation, a new subspecies, not a subspecies, a new variation of animals through genetic engineering. So animals that are endangered also are being cloned to preserve the animal diversity. And after that, we could also have a salmon that is fast growing, which is genetically modified. You have a glowing fish also, which is genetically modified as a pet. You could go even for a mushroom that won't darken or doesn't produce brown, because once you harvest mushroom, it becomes brown and it's not good for the market. So through CRISPR-Cas, this is the new, actually this is a gene editing, not a GMO. You could produce already a mushroom that will not go brown right away. It's like a slow ripening, delayed ripening papaya. I just want to show you this picture of Bill Gates, who is very much interested also in genetics, which funded our projects on stress-tolerant rice for Africa and South Asia, which it ran for more than 10 years. And this is a project that he was so satisfied that it had helped the South Asia, like those I mentioned, on salt tolerance and flooding tolerant and drought tolerant. Okay, I think it's now on my last slide. I just want to share that my Christian faith and my genetics, this is what I want to say, that once I see my faith and my field of expertise on the field of genetics, I could really appreciate the beauty of our creation. You could see from heaven above, you could see God, but once you go to the micro level, up to the DNA level, you could see the beauty of God. And you could see even in Genesis, which I'll be mentioning later, that kind will produce kind. So species will produce the species. So genetics is so accurate that any changes, it will allow some changes through mutation, but most of the mutation will die if because they are deleterious. Or else, if there's some mutation, there is a restriction enzyme that will cut it and cut it out. Parang niluluwa niya, ina-abort niya, and maintain again. Meron ulit na mag-change ng DNA back to its original form. There's a mechanism there. That's why you have restriction enzymes and the polymerase chain reaction. The polymerase, it will create again the new. There's a copy. There's an editor there in the replication. So any mutation they will edit out and repair. And you know, there's I read the book of Darwin's Origin of Species. It's a beautiful book, but it doesn't say about evolution. It only, Darwin was awarded for his on botany and zoology, but not on his study on evolution, because he doesn't mention about evolution. He talked about natural selection, but there will be some changes a little bit, but it will not jump from one family to the next. It will be dog, it will be the same as dog. And he did mention that the dog will become a bird or a dog becomes a cat. So those are the beauty of how Darwin explains those things. So as I mentioned, nucleotides, we have 80 CG, 4 letters alphabet. Every human being, every life has all of these 4 combinations, and it's just a matter of recombining them to produce a species. If there's only 4 codes in life, it means there's only one life. There's only one designer. There's only one engineer of this one, which is God. It's very obvious in this one. And I believe that God, the first creation, was perfect. But after the sin of man, after the fall of man, the disease was there. And I think he spread also the good things. It will not come from one thing, it spread. Not all crops they have all the desirable traits. So he spread the undesirable, the good traits, and it's up to man to recreate the creation. So that's why you say man will work to put back the new creation. So that's where the genetics will come in. He will get all the spread, and by hard work, labor, studies, and put back a better crop, a better animal for human needs. I think this is my second to the last slide. I'll be, what are the current issues in my field that challenges my Christian faith? This is very critical. So of course, the number one is evolution. There's a lot of study now of systematic biology. A lot of my friends are geneticists. They are very good in studying, and they believe that everything is evolution as you're reading the textbook. They have several studies. They have seen some evolution, but they are, as usual, they are extrapolating it to make evolution more attractive. So as I mentioned, kind by its kind, I could not get anything transition in between. So that's the question. There's a big debate on this one, but I believe, and I'm successful in my field of genetics by not believing on the evolution that they're talking about. There's an evolution from one, some changes, maybe changes, the hair become longer or the beak becomes shorter, but it is not changing into from a bird to even from a chicken to an eagle, or an eagle to a swan. So it has some boundaries in between, because it's really biblical to say it's kind by its kind. GMO and genetic manipulation, they say we are playing God. Actually, we are just recreating. We are given the stewardship of nature, and we are just using his tools, God's tools, to put back together the good things together. And they say, what God created, don't tamper it. So usually that's what they are saying. But of course, you say, as I mentioned, God put us in this world as steward, and we have to repair what is broken because of our sinfulness. And they also say, geneticist, you're a geneticist and you believe and you do not believe on evolution. That's not compatible. No, it's more compatible. All the more you will see, you appreciate genetics, all the more you will appreciate the creation of God. And you will have also uh, I always say that be a Christian scientist and have a testimony, you don't need to argue all of these things, just show that what you're doing, and have a good testimony in your research, in your field. You do something good, and that will speak much than arguing about all this about the science of evolution and all the stuff. Again, I just want to share to you my last few slides. I want to share that God said in Genesis 1:11-12, God said, let the earth sprout vegetation, plants, yielding seeds and fruits on the earth, bearing fruits after their kind, with seeds in them. And it was so. The earth brought forth vegetation, plants, yield seed after their kind, the trees bearing fruit, with seed in them after their kind, and God saw that it was good. So consider the heritability. You could see here the continuous of life as God mentioned. And you could see here also in Genesis 1:29-30, then God said, behold, I have given you every plant yielding seed that is on earth, on the surface of the earth, and every tree which has filled. I think these are the same as I have been reading the other slide. And again, this is my last slide. I will say in Genesis, in Galatians 6:7, do not be deceived. God is not mocked, for whatever you sow, whatever one sow, that he will also reap. So I'm just giving an example that what you sow is what you reap. So Christianity actually is all about the grace of God. He gave us good things which we could not, we could never have earned in our own. You could see here that also we have, we deserve nothing but the consequences of our sin, of our mistakes. So we sow our sinfulness, our mistakes. We sow our being greedy, we'll reap what we sow. But God gave us solution that is in Christ. So then we know that Christ bore the consequence of our sin, and that is his solution. And by dying on the cross, and if we believe on the Lord Jesus Christ, who is the creator of everything, and he will give us an everlasting life. My message here is we have to appreciate. I appreciate my field in the field of genetics. I appreciate life. I appreciate nature. And I believe there's a creator up there who is looking unto us. At the same time, he is a holy God who knows that what we are doing, and we know that we are sinful enough. We deserve all that we have, and we don't deserve anything from him. But his solution is the Son, Jesus Christ, who bore our sin, who died on the cross. And if we believe in him, and if we repent from our sin, he is there to accept us and give us an everlasting life. Thank you very much, and God bless you.